We know that beta-catenin stabilization in the dorsal side of the embryo is required for the formation of dorsal structures. So what is beta-catenin doing in these dorsal cells? Recall that beta-catenin is a transcription factor. In the dorsal cells, beta-catenin binds to the promoters of genes that are required for axis formation. These include the genes siamosis and twin, which are also transcription factors. The expression of these genes is usually repressed by a second factor, TCF3, which becomes an activator once it binds to beta-catenin. Once siamosis and twin are expressed, they bind to the enhancer regions of other genes. In the vegetal cells, these two transcription factors, siamosis and twin, work together with vegetal transcription factors to help activate endodermal genes. In the organizer region, these same transcription factors drive the expression of genes that are required to activate organizer genes. Some of the genes regulated by these transcription factors are genes that are required for the specification of the dorsal mesoderm. An example of that is the gene glucosid. Other genes are required to induce the ectoderm to become neural, and these include the paracrine factors chordin and noggin, which we'll look at in more detail later. It's important to know that even though the transcription factors siamosis and twin are also present in the vegetal cells and contribute to the specification of the endoderm, the presence of the VEGT transcription factor in the endoderm or the future endoderm prevents the organizer genes from being expressed outside of the organizer area. Besides beta-catenin, another factor that is required to activate the organizer genes is a transcription factor that is activated downstream of the nodal signaling pathway. In the previous lecture, we saw that the maternal factor VEGT activates the transcription of the paracrine factor nodal, which functions to specify the mesoderm. Nodal expression is also activated by beta-catenin. So in regions where VEGT is expressed and beta-catenin is present, the levels of nodal are highest. In regions with less beta-catenin, the levels of nodal are lower. This is what's shown here in part C of this figure. The paracrine factor XNR is the same as nodal. So a gradient of nodal forms, and depending on this gradient, different genes are activated. Where the concentrations of nodal are low, the ventral mesoderm forms. At intermediate nodal concentrations, the lateral mesoderm forms. While at the highest nodal concentrations, the dorsal mesoderm, which is the nodal cord, forms. So the cells that make up the nucleus center, which is located more vegetally, go on to give rise to the endoderm. The cells of the organizer become the dorsal mesoderm, the nodal cord, and during gastrulation, these cells will be migrating below the dorsal ectoderm, which becomes the nervous system. The organizer is made up of 4 different cell types: pharyngeal endoderm and head mesoderm, which are the first tissues to be internalized during gastrulation, and which are going to induce the formation of the anterior portion of the brain, the forebrain and the midbrain. We also have dorsal mesoderm, which is the nodal cord, and that will induce the hindbrain and the trunk, or the spinal cord. And those are located more posteriorly compared to the forebrain and the midbrain. And finally, the dorsal blastopore lip, which induces the formation of the tip of the tail of the amphibian larva, which is the most posterior dorsal structure. So the organizer serves 4 major functions. It can self-differentiate into dorsal mesoderm, so it will become the tissues that are derived from dorsal mesoderm. It can dorsalize the mesoderm that's around it into the type of mesoderm that gives rise to the muscles of the back, and that's known as paraxial mesoderm. And it can also dorsalize the ectoderm and induce the formation of the neural tube. And finally, it can also initiate the movements of gastrulation. So how does the organizer dorsalize its surrounding tissues? How does it induce the formation of neural tissue? Well, the organizer produces paracrine factors, which are important for allowing the ectoderm to become neural. In an experiment in which a nude dorsal lip tissue, which is the organizer, was placed on one side of a filter that could allow the passage of paracrine factors but not cells, and a nude gastrula ectoderm was placed on the other side of that filter, the ectoderm became neural tissue, which is what we see in this image, where you can see rudimentary eyes. So if the organizer, the notochord tissue, doesn't migrate under the ectoderm becomes skin instead of nervous system. Now, because of this, scientists initially believed that the factors of the organizer were inducing the ectoderm to become neural tissue. But it turns out that the factors secreted by the organizer are actually inhibitors that protect the ectoderm from signals that would induce it to become epidermis. So what that means is that the epidermis is what's induced to form, not the nervous system. So that also means that the default state of the ectoderm is actually neural. In order for skin to form instead of neurons, paracrine factors have to activate the expression of skin genes. These genes code for paracrine factors of the bone morphogenetic protein family or BMP family. These BMPs are initially expressed throughout the ectodermal and mesodermal regions of the late blastula, but their expression is blocked by the organizer-specific transcription factors zymosis and twin. So the expression of BMPs becomes restricted to the ventral lateral region. However, since these are paracrine factors, they could still diffuse and affect the ectoderm that should normally give rise to the neural tissue. To prevent that, the organizer secretes molecules that block BMPs from functioning. Where these inhibitors are not present, so far from the organizer, the ectoderm is induced to become epidermis of the skin by the presence of the BMPs, because BMPs induce the expression of genes that are needed to form the epidermis, while preventing the expression of genes that are needed to form the nervous system. But near the organizer, the BMP inhibitors produced by the organizer protect that ectoderm from BMP signaling, and this allows the nervous system to form. So BMPs induce naive ectodermal cells to become epidermal, while the organizer produces substances that block this induction. And these BMPs also act as morphogens in the mesoderm. In the absence of BMPs, a dorsal mesoderm or notochord forms. This tissue will play a role in allowing the nervous system to form, as we just mentioned. At low levels of BMPs, the intermediate mesoderm forms, while at high levels, the ventral mesoderm forms. So the organizer acts by blocking the BMPs, and three of the major BMP inhibitors secreted by the organizer are noggin, chordin, and follistatin. So let's look at what happens when these BMP inhibitors secreted by the organizer are absent, or when they are present at levels that are higher than normal.